Hello, hello, hello. How are we today? Are we stressing out about DAA yet? No. If y'all will appease me, write down on one of these sheets of paper what three things, or actually just in general, what you felt very, very confident about on this biochemistry exam and what you felt not confident about from this last biochemistry exam. Very informal. No one will read this but me. We all got hundreds. No,
I wish we did. But I wasn't.
Or are you doing your own thing? Are you doing your own thing back there? Cool. Overall, did we... Cool. All right. So, chapter 5, cell signaling. Let's turn this thing on. So the last things that we covered were enzymes. So we started proteins, we got enzymes. Now we're going to get a little bit more specific with cell signaling. So it's going to be more of how cells are able to communicate with each other and initiate actions within the cell. So this is going to be kind of moving away from the chemistry and more into the biology. So for those of you who are excited about that, good for you. But if you're like me and you like chemistry a little bit harder, a little bit more, also good for you because I'm teaching it. I'm joking. That was a joke.
Candy today?
Say again?
Are our candies today?
Yeah, I forgot. I left it in my truck. I'll go get some at the break. I'll go get it. Good reminder. Good reminder. Candy, good. All right, cell signaling. So all this is talking about is how, like I said, we're going to be initiating actions within our cell. So this is very microscopic, very zoomed in, what's happening on the cellular level. Just take that into mind. All right, I'm going to do just a little bit of reading, intro style. So chemical communications are the major ways by which our cells communicate with each other. Cells receive signals both externally and internally. So we have our sensory neurons receiving information, but we also have chemical signals, things binding to receptors, what have you, on the inside of our bodies as well. And the biggest thing is that we use chemical messengers, specifically our neurotransmitters, hormones, toxins, pathogens, what have you, to do these communications. And this signal, it creates a signal, and that's propagated by a sequence of more chemical reactions. Still, it's just biology. It's not a lot of chemistry. What happens, though, is in our body, it's trying to be as efficient as possible, right? So if something happens at one cell, it's probably going to want to create a global response within our body, too. And what happens is the hormone or neurotransmitter will bind, and then that'll bind, and then that will create kind of a flood, mass information created after that to make things happen quickly, right? So if we just have one thing acting on one thing, and then one thing other happens, it's going to be very slow. So what the body has learned to do is to take one signal and just amplify it to the max. Right? It'd be like me talking like this. Amplify your sound to get a bigger response than just to do it without amplification. It's faster, more efficient. Cool. So how it does it, we need to know this general formula. This is just the very basic how this works. So we have some sort of signal. If you want to think of it like in your if you ever took a communications class, like this is how you communicate efficiently or at all. You have your signal. Something's got to receive that signal. So you have your receptor. Signal binds to the receptor. Then you have your transducer, which interprets it, leading to our effector and then our response. So we could say that me talking right now is my signal. Your receptor would be your ear. Right? It receives the information, goes to your transducer, which is your brain, right? Then it goes to your effector, which would be your hand, and then your response would be writing the notes, or writing down this example, all right? Or you could say signal on the level that we're about to do here, like signal is a hormone. Hormone binds to a receptor, the cell nucleus, or whatever, determines what it needs to do with that, creating our effector and our response. Very simple. Need to know that order. He asks what that order is every time. Did he ask y'all the S plus E, SE? No? I tried. But we knew it, right? But we knew it. Cool. Yes. Cool. So we need to know that order. And there's different ways of the signal transduction: autocrine, paracrine, endocrine types. We'll get to that here in a second. Here's a more practical response. Or example. We're going to skip it because I explained it wrong last time. So we're just not going to. No, we'll do it here. So stimulus are going to be our secretion. We're going to have our sensory cell. Sensory cell is going to receive that information. Then it's going to go to our signal transducer, which is going to interpret that information. It's going to create an effector response, which will act on our target cell. Very, very simple. So, all right, so mechanism. Actually, let's see here. Ligands can be of two types. All right, if we ever see the word ligand, that's kind of just one of those definition words. You can kind of think of that as like our substrate that we talked about in previous chapter. So our ligand is what is going to bind to our receptor. So even if we wanted to go back here and change signal to ligand, or in parentheses, ligand, that's where that falls into play. It binds to the receptor.
Andrew?
Yes.
Just to be clear, I had somebody else asking me this and maybe wonder. The ligand can be considered the first messenger?
Yes.
Okay.
Yes. All right. We will get to some of the other information here a little bit later. Let's work on cell-to-cell communication. All right, there are 3 different ways our cells can communicate with each other. So we can have autocrine. What does auto mean? Self, right? So this is just when the cell is talking to itself, right? It's communicating with itself. So whatever the chemical messenger or signal that it sends out, somewhere else within that cell, on that same cell, the receptor is going to be. So this would be like you, I guess, like pinching yourself, or like you doing something to yourself, right? Like you're giving that message to yourself for you to know, for you to do something with. That was a terrible example. I'm sorry. Paracrine. Paracrine. So this is going to act on a different cell, typically one that's kind of around it. So this is very local. So there's going to be our hormone or chemical or signal, however, or ligand is going to be released from one cell, and it's going to go act on an adjacent cell, so something in the same area. And then we have our endocrine. So our endocrine, this is where our chemical messengers are released, and they go into our bloodstream, and then they go have an effect on a target cell somewhere else in our body. All right? So these are going to be like our global hormones, where we're going to have like our fight or flight. Epinephrine is just going to be like dumped into our bloodstream and making a huge global effect elsewhere on the entire body, right? So if we see a lion or what have you, like we're going to need our whole body to turn on that fight or flight and be able to either run away from it or tackle it and destroy it, right?
Quick question. In lecture, Dr. Sarkar was saying that the autocrine isn't necessarily that it's its own cell, but it's its own kind of cell. So that if an A cell, it would affect other A cells, not necessarily itself.
Okay.
Is that right?
Yeah.
Okay, just make sure I understood.
That can, yes. So if that's what Dr. Sarkar said, absolutely. So, and that makes sense too, right? So auto meaning same, so the same type of cell also makes sense. So I guess it could be like something like muscle cells, all muscle cells, or all renal cells. You need to know those different 3 pretty much just, I'm pretty sure just at that basic level, that paracrine is going to act on different types of cells or other cells around it, autocrine either on itself or similar cells, whereas endocrine, we have our whole endocrine system, right? And so those messengers are going to be put into the blood to have global effects on our body. All right, so that is the, I guess, how the messages can be released and where to go. Now we have our receptors. Our receptors are mostly protein in nature, right? So that's why we did proteins for the last 2 chapters up until this point, is because most of our receptors are proteins. So they receive these signals. Many first messengers are chemical molecules. So we have first messengers and second messengers. And our first messenger is just going to be that initial hormone or chemical that binds to the receptor. All right, so just get that covered first. Our first messenger is that first signal. And that can bind reversibly. So let's just underline that it can bind reversibly. All right. So this receptor, ligand, or message binding, it's just like, because proteins, we said enzymes were very specific, right? Enzymes are protein in nature, so it would make sense that our receptors are very specific also, right? We don't want our epinephrine, or we don't want like our, we can say like our acute stress hormones to have the same receptor as our like chronic long-term stress hormones, right? Because you can confuse those types of messages that our body is sending, leading to really non-functional results within our body. So we need that specificity to create specific responses for us to correctly respond to our environment. So it's very specific, high affinity, so it's a high attraction. It's reversible. We need to be able to undo that response, right? We're not meant to be, we as humans, we're not meant to be chronically stressed, right? That leads to increased cortisol levels, increased fat deposition, negative long-term effects. So that's why chiropractic is so awesome, is because when we are getting these patients who are stressed out for long periods of time, right? So we can go in, and with that adjusted thrust, we can actually activate, we can turn off that sympathetic and get them into a more parasympathetic state, thus like reversing that. And so the fact that we need that receptor binding is reversible, we need to turn things on and off. And that's where chiropractic can help. It's saturable. We only have a certain amount of receptors in our body. We can amplify the signal like I talked about earlier. It's got to produce a biological response. So going back here, we start with the signal, we end with response. It's going to bind for a reason, right? That message is sent for a reason. We need something to happen. So that's all it's talking about, is it must produce a biological response, or else it's just wasting time and energy and space in our body doing absolutely nothing. And once we do whatever we're supposed to do, once the action is completed, the body's going to recognize that innate intelligence, and that receptor is going to, or that ligand is going to dissociate from that receptor, thus stopping everything from happening. So we're going to be here for hopefully not 2 hours today. So like me walking in, we start class, we're done with the material, our body recognizes that, y'all recognize that, so we go home. That simple. All right, so this shape fits there, that one doesn't. Pretty simple. We have different types of chemical messengers. I don't think he's going to ask you specifically about these, but our nervous system, we're doctors of the nervous system, yes? So we should probably start familiarizing ourselves with these. So like acetylcholine, norepi, GABA, y'all get that. Starting and y'all's try with DAA. I guess y'all started with cells and tissues. You also have a nervous system with Dr. G at the end of the trimester. We have our endocrine system. Our steroids, our thyroid hormones. Thyroid hormones, they help regulate our, our metabolism. Thank you, that's the right word. Yes, T4 is mostly bound. We just had a test over that, so it comes back. And our immune system, we have our cytokines, histamines, eicosanoids. I'll get all that in Try 3. But those are just examples. So the biggest part here, which is going to be a lot, this is the rest of the chapter really is intracellular versus cell surface receptors. All right, intra meaning what? Within, right? Inside. So it's inside the cell versus cell surface receptors. So for the rest of this chapter, all we're talking about is, simply put, sometimes our first message is going to go and bind inside the cell to the receptor. That receptor is inside the cell, or it's on the outside of the cell. And both of those create different responses, but that's all it is. It's just one response is started inside of it, one response is started outside of it. Cool. Intracellular receptors. So these are cytosolic receptors. All that means is that they're in the cytosol. The cytosol is inside the cell. Also called nuclear receptors. This is typically how you're going to see it, because a lot of times they're on that nuclear membrane within the cell. And the examples we have are steroids and our thyroid hormones. The reason that we have two different categories are based on the polarity or non-polarity of those messengers, right? So our cell membrane is made up of what? Have y'all learned that yet? Phospholipids, right? So phospholipids, it's that phospholipid bilayer, okay, kind of right here. So the phospholipid bilayer has these fatty acid tails that are nonpolar, right? They're just carbons, hydrogens, but they have these polar heads on both sides, correct? Okay? Our steroids and our thyroid hormones are nonpolar. So what that means is that they can just diffuse straight through here, not a problem. They're small enough, they have no charge. If they had a charge, they'd be trapped up here. They'd be attracted to that other polar surface, right? And so since they're nonpolar, they can just go straight through. They don't need any of these proteins. However, for our polar molecules, they're going to be attracted to the polarity on those receptors, right? They have that specific polarity or that specific shape that they're attracted to. So they're going to get caught either here to where they can't get through, or they're going to be attracted to wherever the receptor's at. So that is why steroids and thyroid are intracellular receptors, because they can just go straight through that cell membrane to wherever their specific receptor is at. Need to know steroid and thyroid as examples. So for our cell surface ones, like I said, they have a polarity. They can't just diffuse freely, just because, specifically because they have that polarity. That's why we covered that earlier, so that you can understand what that is and how that affects function. Structure determines function, always, always, always. Cool. So here's just an example of it binds on the outside of the cell and then is going to create a response inside the cell. So membrane receptors. So our membrane receptors are the outside ones. They're not the intramembranous. This is going to produce a second messenger molecule. So the cell receives that first messenger, right? That's the first domino that initiates the sequence. But that message is still on the outside of the cell. We somehow need for that message to get on the inside to create our response within the cell. That's where our second messenger comes in, right? So the first messenger did its job, said, hey, something needs to go on. Second messenger says, I'll take it from here. I'll make sure it gets done. And so that just transfers that message and amplifies it within the cell. Yeah?
So does that mean the nuclear or the intercellular ones, they don't have a second messenger?
Pretty sure yes. Pretty sure yes. I don't know if that's like a always thing, whenever I ask professors an always question, they always say, they usually say most of the time yes. They can never like definitively say one way or the other, but pretty sure yes. Cool. So the examples that we have here are our ion channel receptors, or mainly what we're going to be dealing with are our G protein coupled receptors. That's what the majority of the chapter is going to be, or receptors with enzyme activities. Our sodium potassium pump, or not, or we have channels that can just allow, all right, number one, ion channel receptors. Acetylcholine will bind. We'll ask, yeah, we'll do the question.
On the last page, how are membrane-mediated receptors faster than steroids and thyroid if they have to like start a cascade rather than just diffuse through the membrane?
Cool. So you're asking why the membrane receptors are faster than the intramembranous ones. So we have, great question actually, very good question. The way that we are learning it is that, you can't see any of that. With first distance, so that first membrane only has to go to the outside of the cell, whereas the second one for the nuclear receptor has to go all the way to the inside and find the membrane receptor. Second is faster because this second messenger molecule is going to amplify that signal, thus creating a larger signal, creating a bigger response. So by the time this nuclear receptor gets there, our second messenger has already blown up. It's like it's trending. It's already trending over here, whereas you've got like 3 followers over there and no one knows the message yet. Social media. Is that a sarcastic clap? Does that make sense? You can say no. Please say no if it doesn't make sense. That's a no. Okay. Well, how did you learn it?
Well, just that membrane receptors were metatropic and the other one was ionotropic.
Okay.
So then metatropic had to initiate that one, which initiated that one, which initiated that one, and the other one just initiated. And it was like just happened. So I just thought that would be faster. But that's fine. I understand what you're saying.
Okay.
I get that it amplifies and makes it go faster. But
Cool.
I get what you're saying.
Cool. All right, so our ion channel receptors, what they do, I don't think there's a really good picture on here, but they're, we'll go back a page. So here's an ion channel receptor. Acetylcholine, first messenger, our signal is going to bind to our ion channel, our receptor, right? That's going to, then our transducer is going to interpret that binding. It's going to change our shape, allowing sodium to pass through from the outside of the cell into the inside the cell, and then our potassium to flow from the inside to the outside. So our ion channel is just allowing ions to flow in and out. Very simple. So everything, that's the only response. Binds, transduces it, conformational change to the protein, ions come in and out. Very simple. G protein coupled receptors, less simple. We will get to them. Or receptors with enzyme activities. This would be like our insulin, has tyrosine kinase activities. So we can, some of our receptors are actually enzymes, which can, like for example, insulin is going to uptake glucose from our bloodstream. That's the response. Nuclear receptors, this kind of goes back and forth. Apologize for just going back and forth. But steroid, thyroid hormones, vitamin D3. Vitamin D3 is less important for the examples. It's going to go in, bind to the nuclear receptors. Nuclear receptors are going to cause specific gene expression. So our nuclear receptors are in control of what genes are being expressed or not expressed. And big point that these hormones are slower than our peptide or amine, or just, I don't know why he uses those. You could easily just say the cellular cell surface receptors. It's mediated via signal transduction. It's kind of a big deal. Our nuclear receptors are located within the DNA, not bound to it. Also a big deal. They're located within the DNA, but not bound to it. So the ligands bind to these nuclear receptors. So when we activate them, it causes gene expression via the formation of messenger RNA molecules. Have y'all learned the transcription and translation yet?
Yeah.
Cool. So this is what's going to cause transcription. This is what causes transcription within the cell membrane, so nuclear membrane.
You said not bound to it?
Yes.
It just says bound to it.
Oh, I'm sorry. Bound to it. Read it wrong. Important. Bound to it. Rewind. Localized within the DNA and bound to it. Good reading job. Bad reading job by me. Go back to 4th grade. Right. So in this case, we have our peptide, not our peptide, our hormones, or our D3, or our steroids. They go through the cell membrane. They go through the nuclear membrane, right? Because the nuclear membrane is still just phospholipids, right? So goes on to the DNA. So that's where our receptor molecule is at, is on our DNA. Cause of the change, the effect, the effector is going to be the transcription of DNA to RNA, and then our response is going to be whatever protein needs to be made from there. So whatever gene needs to be expressed, it's expressed. That's as simple as it gets. That's really as simple as it needs to be. Any questions from that? Cool.
I have a question, actually.
Yes.
At what point do the nuclear receptors or those ones amplify the signal? Or do they amplify the signal?
I don't think that there is going to be a second messenger to amplify the signal from the DNA.
It causes one reaction.
Yes. So I think on that one, it's going to be dependent on the amount of that first messenger coming in. So the reason we have nuclear receptors happening, in my opinion, is that we are either lacking proteins or we have too many proteins doing something in our body, right? So we can say we need more protein X or whatever, all right? So we don't have enough, so this is how we're going to make it. So we're going to go to the DNA. DNA has all of our information within our body to make, or I guess not make, things. And so that's going to send mRNA, it's going to take it to the ribosome, protein's made. Perfect. We have accomplished what we need. Or let's say that we are done exercising, right? So we needed more proteins, or we needed more oxygen binding capabilities, but we're stopped, so we don't need that anymore. All right. So we can go into our nucleus, say, hey, we don't need all these, we don't need as much energy requirement anymore. So let's start down-regulating some of that stuff because we don't need it. Answer your question? Cool. All right. Classes of hormones. Over here, kind of just draw imaginary lines here. I don't think he's really going to ask y'all about these. But it'd be kind of cool if y'all did your own research and started looking at these now for when y'all get into future tries. But pretty much anything that's going to have any kind of polarity on it. So our peptides, most of our amino acids were polar, right? Most of them, but all of our proteins are going to have some kind of polarity on them, right? Because it's all those strings of amino acids. There's like a 0% chance that all of them are going to be methionine, valine, whatever. There's only 7. So all of our proteins are going to be cell membrane receptors. Whereas all of our steroids and our thyroid hormones are uncharged and they are nonpolar, so those are going to be our nuclear receptors. And then we have our nitric oxide, which is a huge vasodilator. You'll learn that when you get to physiology. All right. I like pictures. So whenever I see a picture, I like going over the picture first and then going over the words. Is that okay with y'all, or do y'all like words first and then picture? All right. Let's take 5 minutes. I'm going to see if I can pull up a video first because there's like moving parts that are going to be with this. So palpate each other. Get your 5 palpations in right now while we do that.
In this video, we're going to talk about about G protein coupled receptors, also known as GPCRs. G protein coupled receptors are only found in eukaryotes, and they comprise of the largest sensitive compounds to odors, pheromones, hormones, and even neurotransmitters. Characteristic of GPCRs is, the name implies, GPCRs interact with G proteins. They're coupled with G proteins. Now, it's important to talk a little bit about the structure of G proteins also. G proteins, in general, are specialized proteins which have the ability to bind GTP and GDP. In other words, they are able to bind guanosine triphosphate, and the third we call gamma. So all of our G protein of events with the formation of the GPCR will alter. Exchanges GDP. Some G proteins can stimulate activity while others can also inhibit. Now, step 5. Once this alpha subunit activates a target protein, this target protein can then relay a signal. It actually has a few ways to regulate this. One common way out of a few is the RGS protein, which is regulation of G protein signaling, and this can accelerate this step. In this video, we're going to talk about G.
All right, we can just sit back and we're just gonna watch this video. It's like 12 minutes, but it's gonna explain things a little bit better than I will probably, because it's an actual professor. Keep in mind, this is probably gonna give you a little bit more information than probably what Dr. Sarkar is gonna ask, but it's gonna do a very good job of explaining everything, and we'll go over it afterwards, and then we'll probably be done. So hopefully no more than 30 more minutes. Then y'all can go study for bones tomorrow.
In this video, we're going to talk about G protein coupled receptors, also known as GPCRs. G protein coupled receptors are only found in eukaryotes, and they comprise of the largest known class of membrane receptors. In fact, humans have more than 1,000 known different types of GPCRs, and each one is specific to a particular function. They are a very unique membrane receptor, and they are the target of around 30 to 50% of all modern medicinal drugs. In fact, the ligands that bind range from things like light-sensitive compounds to odors, pheromones, hormones, and even neurotransmitters. GPCRs can regulate the immune system, growth, our sense of smell, taste, visual, behavioral, and our mood, including things like serotonin and dopamine. Even now, many G proteins and GPCRs still have unknown functions and is a topic heavily researched. In fact, in just 2012, a Nobel Prize in Chemistry was awarded for research on GPCRs. To start off, let's talk a little bit about the structure of GPCRs. It's impossible to really have a discussion about how GPCRs work without having an understanding of what they look like. The most important characteristic of GPCRs is that they have 7 transmembrane alpha helices. If we have this being our cell membrane, and we have this being the extracellular side, and this being the intracellular side, if we have a GPCR, a G protein coupled receptor, it'll span this membrane 7 times. So let's say it starts here, and we go 1, 2, 3, 4, 5, 6, 7. This is one of the most important characteristics of a GPCR. They have 7 transmembrane alpha helices. Since this is such a unique and interesting structural characteristic, we often also call GPCRs 7 transmembrane receptors. So just to quickly label, this is our GPCR here. As the name implies, GPCRs interact with G proteins. They're coupled with G proteins. Now, it's important to talk a little bit about the structure of G proteins also. G proteins, in general, are specialized proteins which have the ability to bind GTP and GDP. In other words, they are able to bind guanosine triphosphate and guanosine diphosphate, hence the name G proteins. Now, some G proteins are small proteins with a single subunit. However, when we talk about GPCRs, all G proteins that associate with GPCRs are heterotrimeric, meaning that they have 3 different subunits, 3 sections. So I'm going to go ahead and draw this out. The first section we call the alpha subunit. The first subunit or section of this protein we call the alpha subunit. The second we call beta. And the third we call gamma. So all 3 of these together, our alpha, beta, and gamma subunits together, is our G protein. You'll notice that I drew the alpha and gamma subunits with a little tail-looking thing in our cell membrane. And the reason why is because these are the 2 subunits, our alpha and gamma, which are attached to the cell membrane by what we call lipid anchors. Now, the final thing about this picture that I need to draw in is our GDP or GTP. As you remember, the whole point of a G protein is because it binds GTP or GDP. Right now, this protein is inactive, and so it binds GDP, guanosine diphosphate. This GDP binds to the alpha subunit. When this protein becomes activated, and we'll talk in just a second how that happens, it will actually bind GTP instead. So now that we've drawn out our actual picture of our G protein, let's talk a little bit about how our signaling pathway actually happens. That's the whole point of membrane receptors, is that they respond to signaling molecules and ligands, and they respond to the environment. So, as we mentioned before, G protein coupled receptors interact with a wide variety of molecules on the outer surface of cells. Each receptor binds to usually one or just a few very specific molecules, fitting together like a lock and key. So if we pretend that our signaling molecule is a circle like this, the shape in which it should bind to the GPCR should be complementary. When this green signaling molecule binds to our GPCR will actually undergo what we call a conformational change. The shape of this GPCR will change, which in turn triggers a complex chain of events, which will ultimately influence different cell functions. So as we mentioned, our first step here is, of course, the ligand, the signaling molecule, has to bind to our GPCR. Once this ligand binds, our GPCR is going to undergo a conformational change. So let's just go ahead and redraw our GPCR. Again, 1, 2, 3, 4, 5, 6, 7, our 7 alpha helices. Now it's a little tougher to draw a conformational change, but the protein is actually going to look completely different. So here, because of this binding, we're going to have a conformational change. The protein conformation of the GPCR will alter. Okay, so let's just write out our first 2 steps real quick. So step 1, we have the ligand binds to our GPCR. Step 2, we said that we undergo a conformational change. So our GPCR undergoes conformational change. What happens next is, because of this conformational change, our alpha subunit, which I'm going to draw in here, is actually going to exchange this GDP for GTP. So just to keep track, step 3, our alpha subunit exchanges GDP for GTP. So the molecule is swapped out. Instead of GDP, we have GTP. Now, because we have GTP bound to this alpha subunit, it'll now cause our alpha subunit to dissociate and move away from our beta and gamma subunit. Now, once this happens, these two different sections, our alpha subunit and our beta gamma dimer, these two together, are actually going to find a protein in the membrane. It's going to alter and regulate the function of that protein. So we could have another protein, for example, here, that the alpha subunit will find and regulate the function. So let's go ahead and write this out. Step 4. Our alpha subunit dissociates and regulates target proteins. Now, during this step, there are a few things I like to note. The first is that both the alpha subunit and the beta gamma dimer can interact with other proteins to relay messages. We're going to focus in on the alpha subunit because it tends to be more common and more. However, the beta gamma subunits can still regulate functions of other proteins. The target proteins can be enzymes that produce second messengers, which we'll talk a little more about in a second, or ion channels that also let ions be second messengers. Now, as we mentioned, G proteins are incredibly diverse. Some G proteins can stimulate activity, while others can also inhibit. Now, step 5. Once this alpha subunit activates a target protein, this target protein can then relay a signal. As long as this ligand is bound to the GPCR, this process where our alpha subunit dissociates, looks for a protein, and regulates that target protein, causing a whole chain of events, can happen repeatedly, as long as this ligand is bound. Now, how can we actually make this thing go back to normal? Well, step 6 is that our GTP is hydrolyzed to GDP. So our GTP loses a phosphate in hydrolysis and becomes GDP. Once this happens, everything goes back to normal, and the ligand will leave, and everything will go back to looking the way it was, and ready to combine with another ligand in the future. This often happens on its own. Eventually the GTP will be hydrolyzed and become GDP, though our body actually has a few ways to regulate this. One common way out of a few is the RGS protein, which is regulation of G protein signaling, and this can accelerate this step. Now that we actually know the steps to this, let's talk about an example. A very common example of GPCR function in our cell actually involves epinephrine, or adrenaline. This is our fight or flight response. So let's pretend that this green ligand, this green signaling molecule, is epinephrine. And let's pretend that our GPCR is our adrenergic receptor. Once this epinephrine binds to our adrenergic receptor, our GPCR in our body that binds epinephrine, this adrenergic receptor will undergo a conformational change. It will swap out this GDP on this alpha subunit for GTP, and this alpha subunit will now seek out this other protein and regulate its function. And it just so happens that the protein that it seeks out is going to be called adenylate cyclase. Now we have our adenylate cyclase being activated, stimulated by our alpha subunit. And what the adenylate cyclase will do is it'll take ATP, adenosine triphosphate, and it'll produce CAMP, cyclic adenosine monophosphate. So it'll take away 2 phosphates from our triphosphate and it'll make it monophosphate. Once this happens, our cyclic AMP here is what we call a second messenger. So our signal, our epinephrine, goes through this entire process, and the signal is transformed into another signal, this cyclic AMP, which is now inside our cells. And this cyclic AMP will now tell our cell to do other things. For example, is that it'll increase our heart rate. It'll also dilate our skeletal muscle blood vessels. Remember, fight or flight. We need to start running or fighting, and so our muscles are going to have their blood vessels dilate. And finally, all of this process is going to require a lot of energy, so we're going to actually break down glycogen to glucose. Now remember, this is our biggest group of cell membrane receptors. It's a pretty complicated process. Just go over it again. For example, our epinephrine binds to our GPCR. This GPCR then changes its shape and undergoes a conformational change. It switches out the GDP to GTP on the alpha subunit, which causes our alpha subunit to dissociate, which will then regulate another protein, and this protein will turn ATP into cyclic AMP, which is our second messenger, and this second messenger will now tell our body to do other things, for example, increase heart rate, dilate blood vessels, break down glycogen into glucose. Now, other GPCRs in our body, the other 1,000, are going to do other things, but undergo a similar process. So in summary, GPCRs are a large, diverse family of cell surface receptors that respond to many different external signals. Binding of our signaling molecule, or our ligand, to our GPCR results in G protein activation, which then triggers the production of other second messengers. Using this sequence of events, GPCRs can regulate an incredible range of bodily functions, from sensation to growth to even hormone response.
So that's pretty much the gist of it. All of that is in Word form.
In this video, we're going to...
Not that video. Is really in Word form on this page here. And we're about to go over this page. There's a few more steps that it stopped at. But here's that same picture on the left. And if y'all notice too, just kind of look over here. We got Dr. Sarkar, Dr. Sarkar, Dr. Sarkar. So he's done a lot of research into these fields. So he kind of knows what he's talking about. All right, so from the top, first thing that's got to happen, we got to have...
On the last page, it has like thyroid and steroid above it, but I thought those were, like went straight through the membrane to react with that.
So most of the time, yes, but most of the time they're going to be nuclear receptors. I guess in this, there are some times where they act as plasma membrane receptors, but most of the time they're going to act as nuclear receptors. So that doesn't, that's why I don't like the always or what have you, but most of the time, can't answer that question. Okay, so first thing that's got to happen is we have to have our signal for all this to happen. Right, so first thing that we all know for whatever is that our ligand is going to bind to our receptor. So that's what happens. So ligand binds to a receptor. That signal is transmitted to the G protein. All right? So that G protein has the three subunits, right? Alpha, beta, gamma. All that is A, B, C in Greek. So they're really, really creative here. So our inactive form of G protein is bound to GDP. So what happens is when that ligand binds, GTP comes in, and it's kind of important to know like the knocks out or like replaces. It knocks out and replaces GDP from where GDP is bound. It's also important to note that that GDP is on the alpha subunit. All right, so that GTP comes in, kicks out the GDP, and that all takes place on that alpha subunit. And that change activates it. And that activation causes the alpha to release and separate from the gamma and beta, right? So we saw that on the picture. The gamma and beta kind of went off to the side. So that separates it. The alpha subunit can hydrolyze. It has a GTPase. Remember, the suffix ase is just enzyme, right? So that has the GTPase, which can hydrolyze GTP to form GDP, and that's just kind of how it turns off. So when he was saying that there's different ways of turning it off, this is just saying here's one way that it can turn off is that GTPase, when the innate intelligence says, hey, it's time to stop this, that enzyme can turn on and turn off that entire process, which then beta and gamma will come back and reassociate with the alpha subunit. Really exciting, I know. Just bear with me a few more minutes. All right, so the alpha subunit has two types. There's a stimulatory and inhibitory. We're mainly going to be talking about the stimulatory because we want something to happen. But like I said, there's thousands of different G protein coupled receptors. So some of them are probably going to turn off functions within our body, right? So maybe something's going on and we need to turn it off. But the one that we're going to be dealing with, I guess for the most part, is going to be the stimulatory. All right, so now we have our activated alpha subunit. Okay, it's got that GTP on it. It knocked out GDP. This is going to go and activate adenylate cyclase. All right, so over here, this is this big squiggly line. Over here, our adenylate cyclase. That is going to produce cyclic AMP from ATP. So over here, adenylate cyclase is going to convert ATP to cyclic AMP. So adenosine triphosphate to cyclic adenosine monophosphate. We could just say that, going back to the, like we said, we have our signal, receptor, transducer, effector response. Excuse me. We could say that signal, receptor, when the ligand binds to receptor, transducer is going to be the conformational change in our alpha subunit when GTP binds. Our effector is it's going to go affect adenylate cyclase, and the response is going to be the production of cyclic AMP from ATP. Like we could say that that's the whole first messenger system. And so that's why that is called the first messenger system, because it did its job. So now we have this second messenger that it's got its own job to do now. So that's why cyclic AMP is known as a second messenger. I've been told by people not to say camp by like all my friends who were like biology majors. So I was the friend who just said it to piss them off. So maybe I'll be nicer, better people than me with your friends. Thank you for the pity laugh. All right, so cyclic AMP is what we're dealing with now. And what cyclic AMP does is it converts an inactive protein kinase to active protein kinase. So the substrate for protein kinase is cyclic AMP. It's also known as cyclic AMP dependent protein kinase, or pKa. So what happens is this cyclic AMP is now considered a substrate, binds to the receptor on protein kinase. The function of protein kinase is to cause protein phosphorylation. What are we doing when we phosphorylate something? We're just adding a phosphate to it, right? We're just transferring phosphate. So that's all it's saying, is that cyclic AMP is coming over and putting a phosphate on, or that protein kinase is going to put a phosphate on a protein. And so whenever we typically put a phosphate on something, that's kind of, it's another way of saying like we're activating it. Like we're labeling this thing for something to happen to it later. Cool. The inactive form, so kind of going back a step, the inactive form of protein kinase contains two regulatory subunits. It's got two R groups that are the regulatory ones and two catalytic subunits, the Cs. So 2 Rs, 2 Cs. Cyclic AMP binds to the regulatory ones and removes them. So cyclic AMP binds to the regulatory ones and removes them. So that leaves our catalytic ones, the ones that are going to have that protein phosphorylative effect, are now free to do whatever they want, right? So that cyclic AMP is like the key to the handcuffs, releasing that enzyme to do whatever it needs to do. Substrate protein is dephosphorylated by another enzyme, that removes the phosphate group and is known as protein phosphatase. Series of protein phosphorylation, dephosphorylation reactions can happen. Okay, so I guess that protein phosphatase can take off. This is where I always just like lose that one spot right here. Substrate protein is dephosphorylated by another enzyme that removes the phosphate. So I guess there is another enzyme, protein phosphatase, which can like undo what cyclic or what protein kinase just did. And that's what that line is. I always get tripped up right there. And so you can just have a series of phosphorylation, dephosphorylation. And then at the end, whatever needed to happen, happened, right? So with every one of these sequences, we have to have that biological response at the end. We're doing it for a reason. This signal is stopped when we break down cyclic AMP or inactivate it somehow. And so once we inactivate cyclic AMP, right, so then our alpha subunit, we're probably going to hydrolyze the GTP on our alpha protein, and then it's going to go back to inactive, and then our ligand is going to dissociate.
Say that one more time. Did I hydrolyze what?
So if we have accomplished our goal, right, we don't need to do it anymore.
Right.
So our body understands that, so they're going to stop producing cyclic AMP. It's going to break it down, and we're going to try to start forming more ATP again, so that when we need to turn it back on, we can. So we go back, we break down cyclic AMP, creating ATP. All right, so we can do that, and then our alpha protein, our alpha subunit of our G protein is going to hydrolyze that GTP back to GDP when it's inactive, and then it's going to come back together with our beta and gamma, and then our ligand is going to dissociate from its receptor also. So there's a lot of different ways that we can regulate this, right? So we can break down cyclic AMP, our ligand can dissociate, or we can hydrolyze GTP, and any one of those steps will cause this whole series of reactions to stop. Because you kind of have to have everything, all those sequence of events to happen for that biological response to occur. And that's all it is. It's just understanding the steps for y'all's quiz or it's not a quiz, it's like an in-class assignment. I think he gives you the whole class for it. Like you'll have your books for it, so you can refer to any of it. You'll have your laptops, so don't worry too much about it. And all you'll need to know, and you'll just need to make sure you look over this for the final. But if you watch this video, if you paid attention while we're watching the video, if you just walked with it through this, if you follow me along with this, it's not too complicated. It's really not. It's just kind of a bunch of different steps. But just knowing that binds to the alpha subunit, GTP binds to the alpha subunit, cyclic AMP is the second messenger, protein kinase has regulatory and catalytic subunits. The cyclic AMP binds to the regulatory ones, removing them, allowing the catalytic ones to work, creating the response. That's really the gist of it. Here's some examples of second messengers. Cyclic AMP is the biggest one. Cyclic GMP also. Yeah, probably the biggest one here. Calcium's also a big second messenger. I hate forgetting things at the top here. This is the GI, is the inhibitory. So when our inhibitory alpha is activated, it inhibits adenylate cyclase. So that's going back here to where it says there's stimulatory and inhibitory. So that whole example was with stimulatory. So that inhibitory one will go, and instead of activating adenylate cyclase, it'll inhibit it. So therefore, that's another way to decrease cyclic AMP, ending the response. Cool, and he's got like an actual video with all these actual shapes, I think, that he should show y'all in class. It's going to be pretty much the same thing, but probably more. It's going to be like 2 or 3 minutes long, so a lot shorter. Okay, y'all did see that? Okay. like we said, turning it off, just inactivating cyclic AMP. And y'all can just read this on your own, the clinical correlates, do some self-study, maybe after y'all's DAA test tomorrow. Cool. Questions?
I have a lot, but I have to watch the video first.
All right. We all are free to go. Thank you for showing up the day before an exam.
Thank you.
I never turned the lights back on. Whoops. Yeah.
Bye.
What we call the deformation of the cell membrane, or the cell membrane stretching and being pushed and